most of you. For those of you I don't know, my name is Mike Bagley, and I have the privilege of being Vice President of Programs here at SoundWaters. And I've been even more privileged to have the opportunity to work with the SRI scientists that you're going to meet in a few minutes. And we're really glad you're here. We're glad you're here for a couple reasons. One is to see the great work that the students have done and learn from them, hopefully a little bit. The other is this is the first in-person event that we've had go on 2 years. So it's really exciting to have the energy here and to see everybody not on a screen for the first time, and maybe the first of many. So why are we here? We're here to honor the work of our SRI students. SRI stands for SoundWaters Research Intensive, and the program, as many of you know, was born 2 years ago, summer of 2020, from the idea that there's a lot of students who are interested in, have capacity for advanced science learning, but you can't always access programs like this unless you travel to Woods Hole or Bermuda or the islands. And so we decided, let's create something right here in Stanford that Stanford students and students from the region can access. And so in summer 2020, we did a series of weeks, 2-week sessions. We did it again in a series of end of 2020, summer of 2021. At the end of the program, several students, Lila in particular, said, now what do I do? And so we said, well, we'll start a year-round program. And so we developed this, and this is the end of the first semester of what will be our habit now of having students in the summer doing 2-week intensive programs and then coming to us a couple of days a week during the school year. And so you're here to see their work. Also, frankly, you're here as part of their learning. What we promise these students is they'll do upper level science. And part of being a scientist is being able to present scientific information to a really intimidating audience. We couldn't find them, so we invited you. But we're really glad you're here because this will be very beneficial to them to have you sitting in the audience and to hear what they have to say. I'm going to introduce Tim in just a second, who's going to MC for us. But I would finish by encouraging you to ask questions. There will be time after each of the presentations where you will be encouraged to ask questions of the students, and we encourage you to do that. And either learn more about the work or challenge them to kind of help you learn a little bit more. So that's the deal. I'll introduce the students real quickly. The first two are easy to remember. That's Lila, and next to her is Lila. And there's Lindsay, Mai, Allie, and Owen. Owen and Lila are alum of our summer programs, so they've been with us quite a bit. And so that's the cast of characters for this afternoon. Without further ado, this is Tim Abbott, who's our educator, who has been working with the students all semester. Take it away. Thank you.
Hi everyone. So as Mike said, my name is Tim, and I've been working as an educator with the SRI students, and they've been doing a fantastic job. So I want to first of all congratulate them because they put in a lot of hard work doing a variety of things. So they come to us twice a week. They're out on the boat. They're doing actual science, field science, and then they're coming here and they're analyzing the data that they're gathering out in the field, which is a hard thing to do if you're a high school student. So congratulations, and I look forward to seeing your presentations. And without further ado, our first presentation is Lindsay Taylor, who's going to be presenting water analysis of 3 distinct locations in Stanford Harbor.
Tim.
Right.
I would have built time for it. I would have built time for it.
Although you would have built in 10 minutes.
Yeah.
It just takes a little while to warm up.
Should have put it on the screen.
Tim. There we go. Beautiful.
Okay, hi everyone. Like Tim said, I'm Lindsay, and for my project, I did water analysis of 3 distinct locations in Stanford Harbor. So what we're looking at is 3 different locations, like I said, and we'll be looking at Station 1, 6, and 8, which I will describe individually. And the three water qualities I'll be going over is turbidity, dissolved oxygen, and chlorophyll. Starting off with Station 1, it's located at the end of the River, which is located right downtown. And as you can see around it, there's a lot of restaurants, apartments, there's more restaurants on this side, and there's a lot of construction. And the River, this is the flow of the water, which goes right directly to Long Island Sound. Next is Station 6, which is right in the middle, open water, Long Island Sound. There's just nothing around it, it's just nice open water. That's just a nice sunset. And last is Station 8, which is right next to the wastewater treatment plant, which is right there. And it's pretty bad. And here's a better view. And around it, there's a lot of stores, a lot of restaurants, and more construction. And this is also the flow of the water. So the first quality is turbidity, which is the measurement of cloudiness of water, and it is measured in nephelometric turbidity unit, or NTU. And you can see that higher turbidity means more cloudiness, and as you can see here, this is really gross and cloudy and muddy, and the low turbidity is nice, clear water. And why is turbidity bad? So you can see first that it's this nice clear water, but as it gets more cloudy, this can block a lot of sunlight, the particles can absorb a lot of heat, which can cause rising temperature, and the rise of temperature can reduce dissolved oxygen, which I will talk about soon. And this can also reduce photosynthesis, which means less aquatic plants since they need it to survive, and this can cause bad vision for aquatic life, including predators and prey. And next is dissolved oxygen, which is the amount of oxygen in water, which is very essential for aquatic organisms. And it is measured in this case milligrams per liter, but it can also be a percentage. And shown here, for healthy water, you want at least 4 milligrams per liter. Anything below that, shown here, that no fish can survive. You want to keep the range between 6.5 and 14 because that's pretty healthy, and between 4 and 6.5 is okay, kind of better. And last is chlorophyll, which is the measurement of algae growing in water. And this is one way that chlorophyll produces their algae. This is phytoplankton, and they do photosynthesis because they contain chloroplasts, which has chlorophyll. And the more phytoplankton or other chlorophyll organisms, too many can cause an algal bloom, which is all this gross green stuff, which can be really toxic to land animals, aquatic life, basically anything. Therefore, my hypothesis, I thought that Station 8 would have the highest turbidity and the lowest chlorophyll dissolved oxygen due to the surrounding buildings and waste treatment. And Station 6 would have the lowest turbidity since the water is not contaminated from any nearby land. The chlorophyll would be the highest since it is closer to Long Island Sound. Looking at the first graph of turbidity of the surface, and therefore the surface is not really quite on the surface, usually a few feet below. And shown in here, there is a sample error at first because there's no such thing as negative turbidity, it's just a measurement. And based on looking at this graph, I noticed they were pretty equal along the line. October 13th, I guess there was a storm or something in Station 6, which was the open waters. That's my own theory of what happened. Looking at the bottom, they are a bit different now. Between September 15th and October 13th, it's pretty high, which I will say is a storm again. But looking at it, I noticed that Station 1, which is the red, is pretty high compared to the rest. Looking at the averages of both graphs, it is determined that Station 1 is the highest at 4.13, and Station 6 is the least. The more turbidity, the worse. Station 1 is probably the worst out of the 3. Moving on to dissolved oxygen, which is the surface 2. Like I said before in the chart, anything below 4 milligrams per liter is not good for fish to live, and anything above above 8 is good, in between is like okay. But looking at here, July 7th for station 8, it's right below the 4, which I said no fish can survive. Either sample error or something happened during that time. Looking at throughout the graph, you can see that it is between 4 and 8, which is okay. It could be better, like up in May 4th and May 19th. The water seems pretty healthy. Look at this, or the bottom, this looks a bit better. All the stations are above the red line. And this is better because towards the bottom, this is where all the organisms and wildlife and plants are and fish, so it's good to see that, but it would be better if it was a bit higher still. Look at the average, none of them seem to stand out than the other. They seem pretty equal. The station 6 is the highest, which means it has the most dissolved oxygen, which means it is the healthiest. Station 1 is like between, so station 8 has the least amount, making it least unhealthiest in this case. Moving on to chlorophyll. They seem pretty different in this case with surface. There are a few spikes for some reason, I'm not really sure why, mysterious. And for the bottom, there are some other spikes, but they overall seem pretty consistent. Looking at the average, station 1 is the highest again, which is bad, and station 6 is the lowest, which is pretty good, and station 8 is again in between. Therefore, looking at all the graphs, the healthiest water is station 6. This is where all the fish will love to stay. So just to wrap up what I said before, station 6 has the lowest turbidity, which is good, and dissolved oxygen is the highest, so that is also good, and the lowest for chlorophyll is also good. And you can conclude the most impacted or least healthy water is station 1. Like I said, it had the highest turbidity, which is not good. It was between station 6 and 8, which is okay, it's not the worst, but looking at chlorophyll, it is again really high, making it worse. And turbidity and chlorophyll, this had a huge impact or effect that it had was the amount of nutrients and runoff from the river. It contained a bunch of chemicals, road salt, gas, spill, debris, and this had a huge impact on the dissolved oxygen as well. So what can we do about this? We want to reduce the amount of waste. We want to use safer or less chemicals. We want less chemical sediments and debris to get into this water. We can have a trash collector. A lot of materials can contain chemicals, which can also contaminate the water. Or maybe a filtration system to separate all the chemicals and the water flowing along unsound. Mostly we just want to spread awareness about this because people need to know the effects of poor water quality so they can learn and be inspired to make bigger projects, more ideas, and better inventions to make healthier water. And basically what I learned that there are so many components of water and they all have an effect on our surroundings. It has an effect on wildlife, it has an effect on us. Every factor is really important. It determines if the water is important or not. So it's good for us to know what's going on with this water, if it's healthy or not, so that we can make a solution about it. Any questions anybody has? Okay, go for it.
What surprised you most from your results?
When my hypothesis is that station 8, which was the wastewater treatment, I expected that to be the worst because it's pretty gross stuff. So I really thought that would be the result, but it ended up being station 1, so I was really surprised. Thinking about it, there are a lot more people there, a lot of restaurants, a lot of construction, so I can now see why it can be so much more. So yeah, that's what surprised me. Yes?
How deep was the water when you said you took the samples at the bottom? And was it equally deep for all 3 sites?
There probably were different. I never put that in my project, but there definitely was a measurement. I would say a few meters down. As you shown the average, it did depend on where it was. Anything else? Yes.
So you had a sample from 1, 6, and 8. Were there 2, 3, and 4, and these just for the most interesting, or were those just the 3 samples? you
Yeah,
had?
there was several more, and these were the 3 we actually went to.
Okay.
We didn't test these. We were out. This was just past information, but we did go to each of the stations, so I kind of had an idea of what it would be like.
What it would be like when you chose these 3 ahead of
Yeah.
time? Okay.
Okay, awesome. Great job, Lindsay. And Lindsay is a local Stanford student. She's from Stanford High School. I didn't know. Awesome job. I would like to bring our next presenter, Allie Pinto, up, also from Stanford High. And she's going to be presenting on the effect of rainfall on marine debris. Oh, I'm sorry, you're West Nile. Other Stanford
Good evening, friends. My name is Allie, and tonight I'll be sharing with you the outcomes of my project, which related rainfall and marine debris. Okay, this is a diagram of the water cycle. It may be familiar to some of you. So the water cycle is really impressive because it shows how a single molecule of water travels through our planet and then eventually into the atmosphere through precipitation, evaporation, and so much more. But tonight, the part of the water cycle that I'll be focused on is runoff, which occurs when there's more water than the land can absorb. For example, after heavy rain, if the rain hits an elevated surface, then it will run down to the nearest body of water, whether it be a lake or the ocean or a river. And I want to focus on urban runoff, which occurs more in cities such as Stanford. And runoff tends to occur more in cities because there's less soil to absorb the rain. And this can be very dangerous to our ecosystems, which is what I'll be focused on today. Okay, so there is more litter in cities, therefore when it rains more, then the rain that runs down carries the litter into nearby water streams, which is bad for the fish that live there. Okay, so I decided to center my SoundWaters project on this. My hypothesis was if there is heavy rain on a given day, then we can expect to collect more marine debris in the following days. My independent variable was the amount of rain, and my dependent variable was the amount of trash collected. And that is a pretty picture I took one day while we were out collecting trash. So what we would do every Wednesday, my fellow interns and I, we would take that route right outside code, right here. We would find any trash, we would be collecting trash for about 30 minutes, I believe. And afterwards, I would sort the trash we found into their respective categories. What I found really interesting about trash that I found is that most of it, as you can see, are types of plastics, such as plastic water bottles, plastic bottle caps, straws, bags, and food wrappers. Most of it is plastic, and it concerns me because we know that it takes more time for plastic to break down, which, and until it does, it poses a threat to marine life. These are the results from the 6 weeks we went out and picked up trash. Graph A shows the amount of rainfall of that week, and graph B shows how much trash we collected on each respective week. So the results make more sense if we compare them with each other. The bottom shows the total rainfall there were, and the y-axis shows the trash we collected on the days that it rained. As we can see, there is a roughly positive correlation, which means that as it rains more, then we would have a higher possibility of finding more trash during the following days. In summary, the goal of my experiment was to answer the following questions. Is there a relationship between the amount of rainfall and the amount of marine debris collected from the beach? We collected this data by picking up trash and comparing how much trash we found with whether or not it had rained in the previous days. As these are some pictures I took from the first picture is the amount of trash we collected the day after it rained really. I believe there was a storm, and as you can see, there's a lot of trash compared to the trash we collected after there was no rain for a few weeks. Overall, my hypothesis was proven correct because there is a relationship, a very direct relationship. So this could indicate a need for additional trials because after all, the data was only collected for about 6 weeks. So perhaps if we were to conduct more trials or keep going out for the next following weeks, we'd get a better idea of whether or not there's a relationship. For example, if we were to collect trash during the winter compared with the summer, we would be able to see, hey, maybe there's more trash during the summer because that's when more people go out into the beach. And it's just an idea for a future project. And this is an interesting bottle that we found. I thought it's cool. So what can we do to help? We can stay aware of the weather. If there's a rainstorm, we can be more wary of it so we can go outside and pick up trash from our local neighborhoods. And the best part we can start is in our local community. There's so many events that involve picking up trash from our local beaches or the parks. We could definitely help out. This is a picture of Christina, and that's Leandro. Are there any questions or comments?
I'm curious, what about the tides? Did you kind of take note of whether it's high tide or low tide when you were collecting?
Yes, I actually wanted to mention that, but I didn't have enough room in my project. So I noticed that whenever there was a high tide, we would collect less trash, likely because the water carried away the trash that would have been there if there was a low tide. So, yeah.
Interesting.
Yes?
Did you use a particular statistic to determine correlation?
No, all we did was count out the pieces of trash and
And
compare.
look at the trend?
Yes,
And look at the graphs mentioned?
exactly.
Based on your observations, do you think a lot of the trash was coming from the land or coming from the water?
Definitely from land because the trash we found in the water typically had more mold on it and it was just more degraded compared to the other trash, which were from the land generally was from big pieces of plastic. Yeah.
Given the limited resources in the city, this seems to be an interesting thing that you can take to your mayor as to when she should allocate resources down here to pick up trash. I'll get you into a meeting with her.
All right, thank you, Ali.
Nice job.
Our next two presenters, we have Lila and Lila. So we have Lila DeBano, who is from Darien High School, and we also have Lila Shaftel, who is from Greenwich Academy. And they're going to be presenting on the species composition of Stanford Harbor.
Hi everyone, my name is Lila Shaftel.
I'm Lila DeBano.
And today we're going to talk to you about the comparison between species in the summer and fall programs. bottom right corner of this image is the main trawl point. Why trawl? The trawl method is used to track how population data can alter due to environmental changes. The data collected from trawling exhibits the population difference as a result of climate changes and seasonal transitions. How to trawl? The trawl net is thrown behind the engine of the boat and is slowly let out by two people holding each line. As you can see, this is me and holding the line and letting the net out. The net begins to open up, and once the line has reached a certain length, each is wrapped around a cleat and is held until the trawl is over. The boat moves slowly during the duration of the trawl. Once it has been 10 minutes, the people holding the two lines will unwrap the lines from the cleat and pull the net back into the boat. Now that the net is back on board, it is opened at one end to release all the organisms into a tank. The organisms are held in the water to be able to be examined safely.
So as a member of the SRI summer program, this is what we collected over the summer. So the summer trawl data showed a high amount of porgies and comb jellies, as you can see here. This is the porgy, and then this is the comb jelly. And as you can see in the graph, they overwhelmed the amount of other species caught, and this data showed an abundance of the porgy and comb jelly species in the summer is extremely high relative to the other species that were caught. And to put it in perspective, in total we caught 1,812 of the porgy, which is the blue mass right here, and 400 of comb jelly, which are the red part right here. And in total we caught 23 species. And also the leading groups, which are the porgy, the comb jelly, the common silver snail, and then the silver sides. This made up 96.6% of the total biomass that we caught over the summer. And then here is a graph of the less abundant species. So without the silver sides, the porgy, the comb jelly, and the common silver snail. And here, this is only 3.4% of the total biomass that we caught, and you can see it's much greater variety because the other four really overwhelmed the rest of the data.
The population of the porgies and comb jellies is eliminated from the trawl due to the migration within the south for more suitable temperatures. The fall data in the trawl demonstrated the species diversity of the summer sound ecosystem was almost twice as diverse as what was caught from the fall sound ecosystem. The data shown for the grass and sand shrimp is the polar opposite to the porgies and comb jellies from summer to fall. The population of the grass and sand shrimp was minimal but spiked in the fall. Here are some of the photos of the organisms that we caught in the trawl from summer to fall. Small juvenile porgies tend to stay in shallow waters in the summer, then move to deeper waters in the fall. This was found in the comparison between the summer and fall trawls due to the quantity difference in porgies, comb jellies, and sand and grass shrimp. The amount of each species dropped dramatically and demonstrated marine organisms need to migrate to different areas of water. This migration is to fit their temperature needs, especially during seasonal changes. Due to the fact that the number of animals in the fall compared to the summer caught was substantially less, we can see the sheer power and effects of climate change.
And also, as a member of the SRI summer program, this also makes sense because a lot of the porgies that we caught out of the 1,812 were small, which makes sense because they would be the adolescent population of the summer. For comb jellies that we caught, which can be seen right here, they prefer warm water and survive better in it. And this is because the warm water helps jelly embryos and larvae develop much faster. So the comb jelly's reproduction happens more efficiently in the summer due to the warmer water. Also, the population booms in August, and some studies have shown that the adult population of comb jellies sometimes eat the younger population, and this is to sustain the sufficient amount of nutrients needed to survive until the summer. And given this information, our data would make perfect sense, because in the summer, the comb jelly population made up 15.9% of the total biomass that we collected, while in the fall, we collected none, and this would match up with the fact that in the summer, the population spikes, and then in the fall and winter, as the temperature decreases, so does the comb jelly population. Also in the summer, we collected very few shrimp in the deep waters with the trawl net, while in the fall program, the grass and sand shrimp made up a large percentage of the total biomass, with a total of 62.4% of the organisms that we collected were shrimp. This data also makes sense with their life cycle patterns because grass shrimp specifically move to shallow, warmer waters during the summer, and this is where they reproduce and lay eggs, which also, another fact, though this isn't part of the trawl data, during the summer program when we went wading in the shallow water, a lot of the shrimp that we collected, because of their transparent bodies, you could see the eggs in their stomach, which makes sense because it was the reproduction season. And also, during the colder months, the shrimp move to deeper water, and this is because they can survive there due to the more stable temperatures at the greater depth. And all this is supported by the data that we collected. So given this information, we know that comb jellies do thrive and reproduce better in warmer water rather than colder. And because of the data that we collected from the trawl nets, we also know that this applies to the comb jellies in the Long Island Sound. So this information could be very useful for future years of sound research, because instead of comparing the comb jellies from the summer to the fall, an interesting project would be comparing the comb jellies collected from summers to future summers. And this is because as comb jellies do increase, their population increases as the warmer water increases. It would be interesting to see, as in the future years, if the water temperature increases, if the amount of comb jellies collected would increase with it.
For years to come, the shrimp collection data can be analyzed to see how warming waters can affect the migration patterns and life cycles of the shrimp. Using both the trawl net and wading on the shore, the quantity of shrimp can be analyzed compared to the month they are observed in. This can then be used to interpret and analyze if the warming water is changing how long the shrimp are staying near the shore, or how many of them are migrating to deeper waters in colder months. However, there is still a large area for research with this data. Thank you. Thank you.
Oh, does anyone have any questions?
Can you guys go back to the slide where you found all the animals and tell us what they were?
Yes. This one?
Yeah, I'm just not sure what they are, so if you could.
So this, we caught this on the last trawl net that we did. This is a summer flounder. And you can tell because the eyes are on the right side of the body, and there's also less distinct circles on the inside. This is a spider crab that we're holding, and then it's very hard to see, but in the other Lila's hand, she has a pipefish, which is right there. It's like a small, it's actually cousin with the seahorse, which is in my hand.
You can also see a spider crab up here as well. And in the middle, you can see, I think that's you holding a seahorse.
And then also on the right, it's hard to tell, the quality's not very good. That's actually the seahorse again. It's a bad angle, so it looks like seaweed. But that's a seahorse, and then that's also an example of what the trawl data looks like once you bring it in. We put it into these clear containers. So there we have what we caught that day. We have a small spider crab, a young spider crab, a seahorse, and then that also is a sea sponge. It looks also not very noticeable because it looks like sponge, but that's also a living organism, and sometimes you find worms in them too. And then this is the one picture that's not from the fall data. This is from the summer program, but I thought I would add it because it was one of the best finds that we found. So that's a baby squid right there that we caught on the trawl net that we did off of the schooner.
Awesome.
Yes.
When you're trawling behind the boat, how deep is that net? How deep in the water is it?
I don't know. It's a pretty big net. Here, let me see. I'll go back to it.
And I guess the question, ultimately, whatever depth that was, would you have collected just totally different types of fish, do you think, and so forth, if it was closer to the bottom or closer? Is it very depth specific what you were catching, do you think?
The net reaches the bottom, since it's not that deep where we're trawling. It's around 20-ish feet, so it's pretty shallow. So the net reaches the bottom, and it's basically sweeping
the whole bottom, okay.
And also, this is like a small diagram. So here, this is the trawl, like they're called the trawl doors, but those are basically weights. So when we cast the net, we throw the buoys first so that you can tell where it ends, and also to warn other boats. And then after that, we lower the net in the trawl doors, and then as we continue to let it farther out, the trawl doors sink all the way to the bottom, and then it's hard to tell here, but they're flat and they're heavy, so that when you drag it along the bottom, it holds the net down so that it stays on the bottom and collects what's on the bottom, and then it also collects all the organisms that are towards the top.
Did you get any sea stars?
We did not.
No.
Because at one time they were in the levels on the shores of SoundWaters. Yeah.
Do you get much garbage down here?
We don't usually get much in the trawl net. The times that we did it, I don't think we got any in the trawl net. that's usually because the nets, or the net had the holes in it are relatively, in some places they're relatively big. And also because we're so far out, trash would usually be on the surface. And we usually collect what's underneath the surface.
All right, we have time for one more question.
I was just gonna say, what is the porgy?
Porgy looks like a generic fish. I
We have a picture.
think we have a
It's
picture
up
of
there.
it.
It's
Do we?
Yeah.
Oh, here's porgy, yeah. These are juvenile porgies, so they're about that large, but full adult porgies can get up to roughly that big.
We didn't catch many of them.
Yeah, we didn't catch them
Because we caught it during the reproduction season when they were babies. Thank you.
Awesome job, Lila and Lila. Our next presenter is actually from Stanford High. That's Mai Mai. And Mai Mai is going to be presenting on the salinity variations that we see at a few different sites in Stanford Harbor.
All right, take it away.
So here we go. Salinity variations at the 3 different sites, 8, 6, and 1 in Stanford Harbor. Okay, so let's begin with hypothesis. For my hypothesis, I hypothesized that that with more rainfall, there will be less salinity. So if you have more rainfall, that means lower salinity. If you have less rainfall, higher salinity. But let's go into other external factors like weather events, general landscape, and humans. So here we have, let's begin with climate change. So with climate change, we know the world is getting hotter, all right? So it may be just like a little bit, but that can still change the salinity. So for the temperature change, there's the water cycle that Ali discussed before, that will speed up the evaporation process, the condensation process, and then eventually the precipitation process, which is where the rain comes in. With more rain, it lowers the salinities, especially on the top levels of water. With general weather events, it can be assumed that when weather events do happen, it can be more intense, but not necessarily more frequent. Moving on to the food chain and creatures. Why does the food chain and creatures need salt? Now let's take for instance, we have like a fish living on the bottom level of the ocean. There's more salinity down there because salt is dense, so it will start going down and sinking to the bottom. With that, if we were to take a shallow living fish where there is less salinity, taking temperature out as a factor, which is another one, but we're going to focus on salinity. So if we were, for instance, to switch the locations of those two fish, that would be not very good for the fish because they are used to either one level of salinity or the other. If they're interchanged, that can be, no. Okay, so human impact. Let's think. Humans, what are we standing on right now? We are standing on this hard surface. Rain cannot penetrate this. It will maybe roll off the roof and do that, but it cannot generally go in this area, which would have previously been able to absorb this water. Now, if it can't absorb this water, that means the acceleration rate for it to go to these bodies of water nearby is going to be faster. And if it is, for instance, left out in the sun, it's going to evaporate. It's going to evaporate more because this water that isn't able to seep into the ground is going over there. For instance, like the harbor. Okay, so later on I'm going to mention wastewater treatment, the freshwater input that gives, and then tidal movement at Site 6. Okay, here we go. Lindsay, I'm sorry. Okay, here we have materials and methods for salinity, which is measured in PDT, or parts per thousand. We would use either the datasonde or the hydrometer. We first use the datasonde, which seems to have a more accurate computer reading, And then we had to use the hydrometer later, which you have to eyeball and estimate instead yourself. There is a picture of me and Lindsay. Lindsay is pouring water into the hydrometer so we can test it. Okay, here are the locations. Yeah, there's a cute little hermit crab. So first we start around site 1, make our way to site 8, go to site 6, and then go to site 1. I'm going to go into more depth what things are here and how they relate to the salinity. Okay, here is site 8. Here, like Lindsay said before, we have the waste treatment, the water treatment center. So they will be dumping more fresh water here. So that will affect the salinity and make it lower because of the freshwater input. Then we have site 1 over here. This is the west branch. The site 8 is the east branch, so they're all going together. Here we are still experiencing water movement, although it is not as drastic as the freshwater input from the wastewater treatment. So here we have, it's going along the River going down here. And then we have site 6. So we have the west branch over here and the east branch coming over here and emptying into site 6. Here we have the breakwater in the Mountain Harbor. So this is also coming up here. Everything here is circulating and mixing together. So this has the most circulation, the most tidal action. So it can be assumed that with all this mixing, it'll have more common results instead of a lot of fluctuation. Here we have a cute little rainfall. Here we have a bunch of spikes over here, but this, as we can notice, is the biggest spike. I will go into more detail later about how this affects the salinity. Here we have down here dates, and then here we have the rainfall in inches. It's like between 4 and 5. The rainwater data was taken from Weather Underground. Okay, here we have the actual salinity. So there's a little color guide up here. We have 8, which is this blue one down here that keeps spiking a little bit. 6, which is the most horizontal of them all, little spike down here. And then we have 1, which is this little yellow guy right here, which is between the middle and the top amount of fluctuation. Here we have the dates on the x-axis, and on the y-axis we have PT. There is a big dip down here, which they all seem to experience. I will discuss that a little bit later. Okay, now here we have surface salinity again, but this is a box and whisker plot. We have site 8 blue, site 6 orange, and then site 1 as this little gray. So the minimum values start from the line down here, so you can see that. And then the maximum values, you can guess. Let's use site 8 as an example. We can use this one up here. So this is the biggest box. So that means it has the most variability going on here. This is the tiniest little range that is happening. So that has the least amount of changing from the numbers. And then this is the middle. Now, what is outlier? An outlier, you see this little circle here. Maybe wondering, what is that? Why is that there? This shows that there is, this shows that there is input of data that is unusual from the common range that we had established before, and that can be assumed from the rain event that happened in the cumulative rainfall. Here we have bottom salinity. This is one. So we have the same color coding before. And instead of like the surface salinity, these are a lot closer together. So over here, they're practically on top of each other. But they start leaving each other a little bit over here. But we still experience this spike. So what does that mean? Again, so with this little thing here, we have, they're a lot more common in size and also on their position here. So maximum values are a little closer, and so are the minimum values. Again, we have the outlier, which is that uncommon rain input. Salinity input. My bad. Okay, in conclusion, rainfall does have correlation to salinity. So that means more rain, less salinity, less rain, higher salinity. Here we have what I said I would discuss later. We have these little circles here. So we have the dips here in the surface and the bottom salinity. And then we have this rainfall, which goes up instead. So these go down, this goes up. So we can conclude with this, that the more rain there is, the lower the salinity will go, even though these are more similar and closer together. Although we can notice that from the color code, yellow is 1 and 8 is blue. Even though 1 is just the river, we have more runoff occurring here and making it lower than 8, even though 8 is generally fresher, as you can see here. See, it's a little bit like that. Is there anything else in this? I don't think so. And then here we have sources and info. But this is like more just like where I got my stuff, so I can take questions now. Christina.
What was your favorite part of the project? What did you find the most exciting in your findings or in the field? Was it spending time with us?
About the salinity. I liked actually drawing the little things and putting them on here. I don't know, it was entertaining.
Yes.
Making a presentation.
Oh. Any more questions? Hello, in the back.
If you were thinking of your next project, have you thought about what follow-up you might be curious about?
I was thinking about either continuing this or maybe moving on to something including the tides. Because I had some other data like temperature and then the tides, but then it just got too much to put on one presentation. So I was thinking maybe I could do that. There is a person in the front.
I have a comment about your presentation and the presentation on turbidity and the impact of the location 8, where
Yeah, a lot of things
the treated plant is, and that was putting a lot of fresh water in. But-
Yeah, it had a lot of similarities, I
Yeah.
noticed. Yes.
Why did you switch from the data sun to the hydrometer?
The data sun had to be returned, so we just looked at that stronger. I would prefer the data
They were
sun.
horrible.
No, it's just an observation of these massive flooding rains that we might be getting with climate change. Seems like it will impact salinity and runoff, and it's just, everything was a good reminder of all the different impacts of what climate change could be.
Yeah, there's many different factors, but
Yes.
they're all still pretty bad for the environment.
They all seem bad, yes.
How can solutions for climate change affect the salinity, whether or not it can improve it or worsen it?
Well, with the heating up of the earth, if we can somehow, if we can somehow like slow it down or even stop it, which may or may not happen if we try hard enough. With the salinity will return more back to what was before, because if it drastically changes now, that's pretty bad for, as I said, for like the animals and the food chain, just everything kind of collapses. Any more questions?
Excellent.
Awesome job, Lana. And for our last presenter, none other than alumni to SRI 0-1, who is also a Stanford High student.
So let's end the night on a positive note and talk about rising ocean temperatures. So my project is a summary of my study of yearly depth temperature in the salmon. Now, we're going to start with my introduction, my methods/materials, my procedure, my results, and my conclusion. But what's this project all about? That's what I hear you asking. That's a nice photo of sunset. So, my research question is pretty simple. Compared to years prior, has there been a significant temperature change at depths of 2 meters or below at 3 separate locations in the sound? The same 3 that we've all been, or most of us have been taking our data from. And I believe that this is an important question because it'll help us, well, it'll help inform us about the exact nature of climate change and rising ocean temperatures on the sound itself. My hypothesis is pretty simple. I do think that there is a significant temperature change compared to 2020 and 2021 at the 3 locations we took. And this is my materials and methods. How did I go about collecting this data? And that's a picture of me. I'm shaky. Like my mind, I used the sonde to collect temperature. I didn't use all the other extra stuff. I just wanted it for simple temperature. But when we had to return the data sonde, I used a Lamont bottle and a thermometer. Now, I'll show you a picture of a Lamont. I'll show you a picture of a Lamont bottle later. So our methods, or my method, is pretty simple. With everyone else, I go out on the sound, and I take either the sonde or the Lamont bottle and the thermometer, and I deploy it just above the bottom of the ocean, bottom of the harbor, which is about 7 meters at most, which is like 20 feet, 22 feet. And then with the data sonde, we just collect the temperature, then we lift it back up and we read it on this crazy screen that it has. Or I would just use a thermometer to test the data I manually got, and then I reported my results. So that's a picture of the sonde. You guys have seen it before. And that's just a screen of all of the data points. Temperature is right there. That is a Lamont bottle. It's essentially just a big tube with 2 rubber stoppers at the end, and it's connected to a rope with a weight. And once you have touched the bottom with the bottle, you just let go of the weight, and it falls all the way down, and it hits a button, and the button releases these 2, and it collects all the bottom water so that you can later lift it up, pour it into a receptacle, and sorry, that's a math class. And that's a picture of me taking temperature. Measured water was the thermometer. Raw data. Let's cut to the chase. What was my data? So this is Site 1. You guys have seen this before, the West Branch. And this is the graph. Now, the blue dot is 2020. This is the 2021 data. Now, because of extenuating circumstances that year, we can only do June through October for 2020. But for 2021, this year, the stuff that I took, we were able to do May through November. Now this would just be the temperature, like you can tell that there's an arc, and this is just a date. We took 2, I took 2 main measurement points, temperature points from each month, so that I had extra data, so that my data would be more rounded when it comes down to it, because I don't want just 5 data points, you know. That's East Branch, and then that is just the same graph, well, not the same graph, it's the same type of graph. And keep in mind that there is the same basic arc that you see, specifically around the warmer months. And then this is the 3rd site, this is SDA6. And again, you can see there's an arc, and it gets warmer on the warm months. Now, results. What does this info mean? So, as you can tell, there's a direct correlation between seasonality and temperature. The warmer the season is, the warmer the depth temperature is. Now, if we go back, it's not that warm, like, it's quite cold actually, but relative to like what it usually is in the sound, it is quite warm, but that doesn't mean it's warming per se. It just means that it's following seasonal trends, which is natural. 2020 and 2021 data points seem to correspond with each other as well, which means they follow the same basic arc. Now, what does this stuff mean? Does this support my hypothesis? It's a well-known fact that when we have studies that that involve long periods of time and data extrapolated from long periods of time, it's better to have many data points because you'll get more precise data. My project, unfortunately, did not have the luxury of 5, 10 plus years of past data to go back on, but that doesn't disprove my hypothesis necessarily. That just means that I have more room to improve, more room to potentially go do more stuff for this project. That's a really nice sunset, by the way. That's pretty nice. In the future, I would use 2020 as a starting point and go 5, 10, good God, even 15 years in the future to see if there is a specific decrease or increase in the temperature. But as of right now, I can't find any real indicator of warming temperatures in the Sound. That does not mean that it's not real. It just means that more data points would be better if we were to draw big conclusions like that. Now, does anyone have any questions? What's up?
Yep, so the, I forget what they call it, but the technical water.
Oh, data.
What's it?
Data.
Data. Did you have to, whatever that was reading out, and then when you had to return that in your next method where you just took the temperature, did you have to compare to see that the data was kind of reading the same way actually, so that you know when you have to, give it back that they would be comparable?
They both read out in Celsius. That's a big one. But the data because it's like really expensive and it's like a real piece of like marine science technology, it's way more accurate than just like a regular piece of like, I think it was like a special cup thermometer that we used, that we had a little cup at the end of it to scoop up some water and we put it in. But the data that we got from both of the temperature collectors, it is very comparable. It's just not as, one isn't as accurate as the other, unfortunately. Anything else?
For any of the studies, do you guys review any literature prior to formulating your hypothesis?
Not necessarily. I mostly formulate my hypothesis based on my own observations, or I guess in this case, not fear, but I would like to know more about this, but I don't want to rely on something that I can't like touch and do, you know? So not necessarily, no.
Mm-hmm.
I don't know about anyone else though.
On something like water temperature, is there a database that you can go back and look at that depth 20 years ago? And
Yes, but for my project, because we're going to 3 specific sites, I didn't want to use like data from like 4 miles away if I'm comparing it with like specific locations, you know. And like SoundWaters has been doing this, like the collection of temperature and like salinity at different points in the harbor, but up until we only started taking data points from these specific sites in 2020. So I only had a pool to draw from 2020, unfortunately. What's that, sir?
Do you see yourself doing something like climate science or something like that in the future?
I know environmental science I like environmental science. I would see myself doing environmental science, maybe not necessarily climate science, because I feel like I would, it's necessary, it definitely is necessary. I just feel like I want something more uplifting. It is necessary. And I am passionate about this stuff. But maybe not necessarily climate itself. Maybe climate adjacent stuff like temperature and stuff like that. Like other stuff. Anything else?
Yeah, thank you. again, thank you everyone so much for coming today, and congratulations to all of you because, again, this is not something that's typical. I wish I got to do this in high school, so congratulations on all of your hard work. And there are refreshments in the back, so please help yourself. And we hope to see you for our spring session. So we will be back with more talks. Thank you.